All right. So today we're going to move on from uh, speciation to something very closely related, which is phylogeny and systematics, which is how do we organize a tree of life that can accurately represent evolutionary relationships between organisms. And there's a number of ways to do this, but it actually is more difficult than you would think, since depending on what criteria we use to say that one organism or one species is closely related to the others, we could get very different trees. And so this is something that's still very much a point of improvement and of research today is how can we make the most accurate tree of life that represents organisms' evolutionary relationships to one another. So first, just kind of unpacking some terms, phylogeny refers specifically to the evolutionary history of a species, and it's based on their relationships by common ancestors. So, organisms that have more recent, in the past, common ancestors are more closely related than those who have more distant common ancestors. And finding these ancestries are based on the fossil record, morphological resemblances, and biochemical resemblances in their body physiology, and then molecular or genetic evidence. Systematics, on the other hand, is similar, but it connects and classifies systems in phylogeny by categorizing and naming the organism. So it specifically has to do with what's the name of the categories and the different organisms. In either case, the fossil record plays a really crucial role, and we talked about this during class the other day, um, because sedimentary rocks created by the deposition of sediments, are really rich sources of fossils. Um, Even so, like we had mentioned previously, it takes a very specific set of conditions to be met for fossilization to occur. So even though we have a substantial fossil record, it isn't complete by any means. So part of establishing this phylogenetic background is looking at paleontology, the study of fossils, since fossils are so important, and they're the most physical examples of change. So, some places where there's permafrost, like you notice in that picture, you can find tusks of woolly mammoths, preserved in this ice. Others, like in the picture of the lake or the riverbed, you'd actually have to dig for them since they're uh, surrounded under many, many layers of dirt and earth. So here are some examples of some different fossils: a scorpion preserved in amber, a leaf impression, a fossilized shell. This large bone I would suspect is a reptile, some petrified wood, and then a humanoid skull. So, we can build phylogenies based on morphological, so body structures and shapes, and molecular, like DNA, homologies or similarities. So, really, the two ways we do this is group organisms based on their similarities in DNA sequence and their bone structures. However, bone structures isn't really necessarily the most accurate if we're looking at evolutionary relationships. Because, for example, this picture right here with the moles, two organisms that have a similar morphology may not be closely related in terms of evolution. So, really, if we want to get the most accurate uh, way of depicting uh, the relationship of species in an evolutionary context, we need to look at DNA. So we need to align DNA sequences based on a common protein, take a common protein, and compare the bases. with the understanding that more bases in common, meaning these organisms are more closely related, because the general idea is that mutation happens at a relatively constant rate over time. And if there are more differences between two organisms, they shared a common ancestor the longer ago. Now, most of this data is analyzed by software. If you're interested in biology and computer science, bioinformatics is a really important field that deals with a lot of that data as well. In systematics, we connect classification to phylogeny. So in systematics, we have that hierarchical Linnaean structure with the genus and the species, like you see on the right. Um, this is not necessarily consistent with evolutionary relationships. It's based more on physical or behavioral traits rather than genetic evidence. So um, while it's very useful, it's got some obvious limitations when we want to compare organisms based solely on evolutionary relatedness. So what we're going to do in class is learn how to build trees like this based on phylogenetic or cladistic data based on derived characteristics or genetic similarities. So one of these pictures, like you see on the wall on the right, um, is an example of a depiction of a possible evolutionary relationship between some different groups or different species or what have you. We call one particularly like this a cladogram because it uh, separates organisms or species based on shared or derived characteristics. So as you see, the cladogram branches according to the time at which a specific characteristic arose along this tree. So if I look at it, this Landsat we call the outgroup. It doesn't have any of these characteristics that are depicted on this tree, while leopards have all of them. A tuna has a vertebral column and jaws, but doesn't have walking legs, amniotic eggs, or hair. Okay, so this is a cladogram based on shared derived characteristics that show the divergence of different species based on the presence or absence of these characteristics. You know, that's useful from a characteristic standpoint, but still isn't exactly accurate as far as looking at evolutionary relationship. Molecular systematics is the most accurate in talking about or trying to discover evolutionary relationship. So this is when we draw trees like you've seen previously, but based on DNA or molecular evidence. And it's based on the principle of what's called parsimony, that the simplest explanation is the best. Or if we have multiple options as to evolutionary lineage between species, the one in which fewest evolutionary events happened is probably the most accurate. So if you look, for example, here are four hypothetical bird species, and we could create these different trees. Okay, and then we would need to look at the data and ask which one represents the most parsimonious tree. So looking at the tree that explains the data with the fewest number of evolutionary events. So as an example here, we have these different options. This first tree would be the most parsimonious. the fewest evolutionary changes overall needed to happen compared to the last one for us to get our perceived distribution of data. We need to remember that these phylogenetic trees are hypotheses. So if we look at these, let's think which is the most parsimonious tree? Well, look at the one on the right. The trait of having a four-chambered heart needed to evolve two separate times in mammals and in birds, whereas in the left, a four-chambered heart needed to evolve once in the common ancestor of mammals and birds. So A would be the most parsimonious tree. Simplest explanation. So modern systematics have kind of shook up our traditional view of how organisms and species are genetically related. As an example, crocodiles are now thought to be kind of one of the most closely related uh, reptiles to birds. Birds and crocodiles share a very recent common ancestor compared to even other reptiles like snakes and lizards. This is all since we've been able to access genetic information a lot more um, certainly. So now that we can compare the entire genomes of different organisms through genetic systemics, we find that humans and mice, for example, have 99% of their genes in common. And 50%, half of human genes, have a close match with those of yeast, which are kind of the simplest eukaryotes. So large portions of our genetic code seems to be very similar amongst even the simplest forms of life. Now, one thing that we want to remember is kind of this idea of a molecular clock, or that small variations in genomes can be used to date evolutionary change, and that the rate of change is calculated um, and then extrapolated kind of like on this data right here. So it says that HIV-1M samples were collected from patients in the early 80s and 90s, and the gene evolved at a relatively constant rate, and that allowed us to conclude that the strain first infected humans in about 1930s. So, what does this assume? This assumes that the background rate of mutation or variation is relatively constant. And at this point, scientifically, there hasn't been evidence at all to kind of rebut that claim. So for most of our uses, that is relatively accurate. And that is a real way that we can kind of look at when traits or evolutionary changes very far in the past have taken place with a similar but more robust methodology. Lastly, it's allowed us to develop kind of this tree of life that has three very separate ancient domains of bacteria, eukarya, and archaea. Here's the prokaryotes, here's the kind of eukaryotes, and it can show us the major splitting points. Each of these splitting points are the most major evolutionary divergences in history that allows us to end up at zero at the modern day with the world that we see.